Hello, my name is Megan Gillespie, and I'm going to be discussing the question, should gene editing be performed on human embryos? Editing the genes of human embryos means removing, adding, or altering sections of their DNA sequence. Scientists are now able to do this using a technology called CRISPR-Cas9, also known as molecular scissors, which cuts a cell's genome at a desired location, allowing existing genes to be removed or new ones added. This technique has enormous potential to cure genetic diseases and improve our understanding of human biology. Since researchers at Sun Yat-sen University used CRISPR to edit the genomes of non-viable human embryos in 2015, the implications of genome editing have been at the forefront of many scientific and ethical debates, raising concerns about the safety of the technique and the consequences it may have on human identity and morality. In this presentation, I will be considering these different arguments and ultimately reaching a conclusion based on the strengths and weaknesses of each viewpoint. The key aim behind editing the genes of human embryos is to eradicate inherited diseases. Many proponents of gene editing state this potential benefit as their main argument in support of its use. Writing in The Guardian, Alex Lee argues that scientific advancements into gene editing and mitochondrial replacement therapy offer nothing but hope, and that we should not allow the fear-mongering over designer babies to prevent advancements such as mitochondrial replacement theory from saving children's lives. He explains how research into genome editing needs to continue, as if we stop all progress into scientific research now, all that will happen is that some of us will continue to inherit often life-threatening diseases. Rather than focusing on hypothetical fears of superhumans or designer babies, the practical applications of genome editing need to be at the forefront of the debate, as many people could have a better quality of life. Looking at the past developments in genetic engineering, Matt Ridley presents a very similar argument. He explains how the consequences of every major development in genetic engineering have been positive, and in some cases have helped millions of people. For example, an estimated 5 million babies worldwide have been born using IVF. This is despite the constant fears surrounding these developments. For example, some feared that genetically engineering bacteria would lead to harmful biological warfare, which has proven not to be the case. Considering this evidence, Ridley concludes that almost nothing but good has come from genetic technology so far, and that we should take comfort from the truly remarkable track record of genetic technologies in alleviating more human suffering than we dared to hope, and encouraging fewer bad outcomes than we feared. A recent development in the field of gene editing supports both Lee and Ridley's argument. In 2017, American and Korean scientists at Oregon Health and Science University successfully extracted an inherited gene mutation from human embryos. This mutation can cause a potentially fatal heart condition that affects around 1 in 500 people. By removing the gene, this outcome can be prevented. This breakthrough has huge potential, as explained by Shukrat Metalipov, who states that, by using this technique, it's possible to reduce the burden of this heritable disease on the family and eventually the human population. It is widely acknowledged that, in their current form, genome editing techniques should not be performed on human embryos that to be implanted, as they are not advanced enough to be safe. For example, in the 2015 landmark study on genome editing, the researchers found that the technique worked properly on just a fraction of the embryos, and often the procedure spliced the wrong gene segment, which led to inserting new genes in the wrong places. In normal embryos, this could lead to new diseases. In a joint statement, Britain's 5 leading medical research bodies recognised these problems, while still expressing their support for the continued use of genome editing in preclinical biomedical research, as well as studies that progress and refine these technologies, as they believe that genome editing technologies may hold significant potential for clinical application in the future. As highly knowledgeable and reputable authorities on the issue of gene editing, their stance sums up the overall argument, which is that research into gene editing should continue, as the technique has the potential to be highly beneficial to those in need of it. However, many concerns have been raised about the safety of the future of genome editing. The dangers of editing the genes of human embryos that are implanted include unintentional edits to the genome, miscarriage, maternal injury, stillbirth, and other harmful consequences that might only present themselves in subsequent generations. Marcy Donofsky, the executive director of the Center for Genetics in Society, believes that far too much is unknown about issues and longer-term health and safety consequences. She argues that the unknown mechanisms of both CRISPR and human biology suggest that unforeseeable outcomes are close to inevitable. In 2015, Donofsky's organisation issued an open letter calling for a prohibition on reproductive human germline modification. The letter, signed by 175 scholars and public interest advocates, explains the dangers of experiments with the human germline and argues that these outcomes could cause social backlash against beneficial uses of genetic technologies. The letter concludes by calling for a prohibition on such germline modification and a robust and broadly inclusive discussion on the socially responsible uses of this and other emerging genetic technologies, which is essentially a ban on genome editing and a conversation about the future of its use. This stance was also expressed by scientists attending the International Summit on Human Gene Editing in 2015. After considering the scientific, ethical, and societal issues surrounding gene editing techniques, they called for a moratorium, i.e. a temporary ban, on making inheritable changes to the human genome. This decision was reached because they believed it would be irresponsible to proceed with any clinical use of germline editing unless the relevant safety issues have been resolved or a broad societal consensus about the use of genome editing is reached. If genetically editing human embryos is possible to do safely, the rights of the embryos need to be considered. The debate around this area focuses on an embryo's inability to consent to the altering of their genome and to the effects this would have on their lives. Most people who write about consent claim that it is important because it protects a person's individual autonomy, which is essentially a person's right to act according to their personal values, free from coercion and with sufficient information to make decisions. Therefore, as embryos are unable to consent, altering their genome would mean denying them of this autonomy, which many believe to be highly unethical. Francis Collins, director of the National Institutes of Health, denied funding for gene editing technologies in human embryos partially due to this reason, stating that ethical issues presented by altering the germline in a way that affects the next generation without their consent is a strong argument against engaging in genome editing. In an interview, Collins raised the issue of consent again, but this time in terms of safety, saying that the question of experimenting on individuals who cannot give their consent arises, as how can you be sure they're not doing something harmful? His argument, therefore, is that we should not edit the genes of human embryos, as they are unable to consent to the potentially harmful consequences. Many people have expressed concerns about how far genome editing will go. Some fear that, instead of being focused on curing or preventing disease, it will enhance human characteristics and capacities. This is known as human enhancement. In the context of genome editing, enhancement means interventions that improve bodily condition or function beyond what is needed to restore or sustain health. For example, improving musculature for patients with muscular dystrophy would be restorative, whereas doing so for healthy people with average capability would be considered enhancement. This is different from therapeutic genome editing, which only aims to cure existing disease or to prevent it. From a philosophical standpoint, enhancement is seen as a quest for human perfection. Michael Sandel, American political philosopher, argues against this aim. He explains that the danger with enhancement and genetic engineering is that they represent a kind of hyper-agency, which is an aspiration to remake human nature to serve our purposes and satisfy our desires. Sandel sees this as a drive to mastery, claiming that this drive may destroy an appreciation of the gifted character of human powers and achievements. Sandel also explains the ethical distinction between therapeutic genome editing and human enhancement, as medical intervention to cure or prevent illness does not desecrate nature but honours it. Healing sickness or injury does not override a child's natural capacities, as enhancement does, but permits them to flourish. Therefore, enhancement is seen as an unethical desire to undermine human nature, whereas therapeutic genome editing would be a celebration of it. So, there are two main arguments against editing the genes of human embryos. The first is that genome editing techniques are not safe, and because we can never control all the dangerous outcomes, we should not continue with research into genome editing. The second is that editing the genes of embryos is unethical, as they cannot consent, and editing the human genome for the purpose of enhancement undermines human nature. Before researching the issue of genome editing, I believed we should edit the genes of human embryos. I saw it as a way of eradicating life-threatening disease and helping to improve the quality of life for, potentially, millions of people. My initial view has been confirmed to a certain extent, with the Oregon Health and Science University's research into the area showing the vast potential of genome editing techniques. This data is highly credible, coming from a reliable and scientific source with qualified authors. Therefore, I have reason to trust their conclusions. Both Document 6 and Alex Lee's arguments give a similar viewpoint to the one I held initially. The difference between them is that Lee's is focused on the future benefits of genome editing, whereas Matt Ridley in Document 6 looks at past developments in genetic engineering to explain why it won't go wrong in the future. I think that both arguments have merit. Lee considers the practical implications of genome editing and the hope it could bring to people with genetic diseases. This point of view is very important to this debate. Ridley's argument that genetic engineering has brought nothing but good is convincing, as he uses lots of empirical evidence to support his point. He also recognises that we need to tread carefully when progressing with genome editing, showing that he recognises the dangers, which is another important factor in this debate. The dangers of genome editing were expressed by many different people, with the scientists at the International Summit on Human Gene Editing encapsulating this argument. They argued that, due to the current dangers of editing human embryos, such as unexpected mutations, continuing with the use of genome editing techniques would be irresponsible. This conclusion was reached by qualified experts from around the world, making their opinion highly valuable. Despite this, I'm not fully convinced by their argument, as I believe that their call for a moratorium on making inheritable changes to the human genome would damage the important progress of genome editing techniques. I now recognise the dangers that come with editing the genes of human embryos, which I was not aware of before, but I don't think that this should affect the research into genome editing. If anything, more research should be carried out so these dangers can be resolved. The arguments that challenged my opinion the most were the ethical arguments, the issue of consent and of enhancement. The idea that we should not edit the genes of human embryos as they are unable to consent to the consequences was a point of view that I had not considered before. However, I do not think that this is a valid reason against human genome editing. Instead of banning it altogether because of a lack of consent, I think that scientists have a moral obligation to edit the human genome in a way that will benefit future generations. Consent, in this case, is only relevant if the consequences are harmful. Sandell's argument that human enhancement is unethical because it undermines human nature is very convincing, as it is supported by highly detailed and developed logical reasoning. Initially, I was unaware of the distinction between therapeutic genome editing and enhancement, but I now agree with the ethical distinction that Sandel makes between the two. His case against perfection provides a clear opposition to human enhancement, and as a reputable authority on the issue, his point of view is reliable. Overall, I believe that we should edit the genes of human embryos. Research has shown genome editing techniques to have huge potential, and I think that scientists should continue to develop these techniques so that they can one day be used safely on human embryos. There does need to be strict guidelines put into place about the applications of genome editing in order to make the process as ethical as possible. The main focus when it comes to editing the genes of human embryos should be on improving or saving the lives of those who would otherwise be born with debilitating conditions, and not on the possibility of human enhancement. The line between therapeutic genome editing and human enhancement is blurred, but with considered and informed debate on this subject, I think that we would be able to determine what is therapeutic and what is not. In order to ensure that the decisions being made have positive impacts on future generations, the safety and ethics of genome editing should be continually discussed as the techniques progress. So, to summarise, my opinion is that we should edit the genes of human embryos, but with caution. Thank you for listening.